Hi.
And greetings, everybody. Welcome to the Evidence and Reasons for the Christian Faith video channel. We're going to talk about John Sanford's book, Genetic Entropy, which was published in 2005. I call that Genetic Entropy 1.0. A lot has happened in the 17 years, and even he and I had written publications that appeared in the secular world. So it was pleasing to see that his work it kind of morphed into something that was able to be published in secular peer review. In light of that, some of the things that he and I co-authored are slightly different than the original 2005 version, added some things, and so I came up with my own version. It's not exactly his, although he and I agreed that I should go ahead and try to write a booklet, and I haven't run the draft yet, because he's going to be a co-author whether he knows it or not. So much of the material is his. And certainly I will invite him to say, yeah, this is where Sal and I agree and where Sal and I disagree. And that's fine. Or maybe we'll just save our disagreements for another. But tonight is a Genetic Entropy 2.0. I would like to thank my distinguished colleague, Dr. Daniel Stern Cardinal. He's an evolutionary biologist. He is a professor of biology at Rutgers University. His input has been valuable, though we disagree on so many things. And my friendship with him has cost me somewhat a standing among the internet creationists, but not among the creationist searchers. I just need to point that out. The people you may hear on the internet that are critical of me, they don't represent creationist researchers or creationism on the whole. And I just want to embrace my colleague, Dr. Sanford Cardinal. He a lot of good inputs. So Genetic Entropy 2.0 does reflect some of the things that he said. I've tried to address it, and I felt he had good points. And so I emailed him and told him that he's welcome to have a separate discussion with me on Genetic Entropy 2.0, because it's not exactly the original 2005 version. So tonight is especially for Paleologos Peter. He's a student of Dr. Todd. He's also a creation searcher. And we have Rebecca, frequent co-host. So Rebecca here has just the nicest personality and a great voice. And she represents people that are probably not nerds like me, and that's probably a good thing, because she can kind of help me if I say something that just like, Sal, can you explain that term or something? So that's what we're going to do today. And I did have some things lined up. Let me see. Rebecca, you have such a beautiful voice. And let me see if I could get this. Well, I'll point out just a few things about genetic entropy while I'm queuing up the stuff. Peter and Rebecca, why don't you go ahead and introduce yourselves? I forgot to put your channels in the description. We were expecting Emery Moyna. He said he might be late, so he still might come, but he's probably rushing home. And I'd like to greet everyone in the side chat. If you all want to say hi to people in the side chat, that's fine while I queue stuff up.
Go ahead, Peter.
All right. Yeah, I'm Peter from Logos. And I've known about genetic entropy for a while. I haven't really gone too far into it, though. And I thought there's really no person better to hear about genetic entropy from than Sal, unless it would be Dr. Sanford. But yeah, so I kind of talked with Sal a little bit about this after my last stream with him. And we'd kind of talked about maybe it'd be great to kind of talk about genetic entropy and his kind of new ideas surrounding it. So generally, I don't really have a firm position on genetic entropy. I'm not really sure. I honestly probably don't know enough about the topic to make a firm judgment about it either way. So I'm really excited to hear what Sal has to say and kind of if he can address some of the criticisms which I've heard of it, or whether those are legitimate or not. So great to be here.
Thanks, Peter. Yeah, and Peter's channel, I love it. Paleologos. He's doing a great job in his field of study, and so he talks a lot about hominid fossils and that kind of stuff related to creation. And so my main channel is Bread of Life. I also have a channel called Examining Evolution, and I have a Bread of Life in Persian. So you're welcome to check out my channels. And Sal is my creation advisor, so I'm really excited to hear from him on this topic. Sal, it was mentioned in the chat that your audio is a little bit weird. It's kind of like crackly, and for me, you're freezing up, but that may be my internet. So, but I think Peter also mentioned that you're freezing up a bit. So I don't know if there's anything that needs to be done, but I just wanted to let you know.
It can crackle when I'm doing other things. Now, if it gets real bad, I'm going to go to my secondary computer, and it has a different audio system. So I really want to thank everyone for the feedback. So please say something, and if it continues to persist. I'm not sure if Sal has ever had good internet. I have good internet. I have lousy computers. So thank you.
Yeah. So you want me to sing until you have it queued up? Are you ready?
I'm ready.
Okay,
Since
good.
you have such a nice voice. This is from Bertrand Russell. Could you read it, Rebecca? And
Sure.
I'll scroll it. Yeah.
Sure. So starting at such.
Yes.
Such, in outline, but even more purposeless, more void of meaning, is the world which science presents for our belief. Amid such a world, if anywhere, our ideals henceforward must find a home. That man is the product of causes which had no prevision of the end they were achieving, that his origin, his growth, his hopes and fears, his loves and his beliefs are but the outcome of accidental collocations of atoms, that no fire, no heroism, no intensity of thought and feeling can preserve an individual life beyond the grave, that all the labors of the ages, all the devotion, all the inspiration, all the noonday brightness of human genius are destined to extinction in the vast death of the solar system, and that the whole temple of man's achievement must inevitably be buried beneath the debris of a universe in ruins. All these things, if not quite beyond dispute, are yet so nearly certain that no philosophy which rejects them can hope to stand. Only within the scaffolding of these truths, only on the firm foundation of unyielding despair, can the soul's habitation henceforth be safely built. Wow, Sal, you know how to find the best quotes. I'm so glad he's wrong.
Well, that is an atheistic viewpoint. And why did I bring that up? This is the ultimate destiny of the human genome. So in the ultimate sense, okay, so the main theory of genetic entropy is the human race is going to go extinct. The question is how. So in terms of the physics, we know it's inevitable. And I know I was on Rebecca's channel, and there's a minor argument over with Dessel Drace. He said, well, humans are going to figure all this out, just like dream on. You know, that some are going to make ourselves eternal or whatever. And he's like, you got a big universe to fight there. And I don't know, there are very few physicists think we'll be able to, you know, correct the universe with power and technology. You can't violate the laws of physics. So All right. So that's why that was Bertrand Russell, who was an atheist. So the thesis of genetic entropy, let me bring this up and see if I can. All right, so Dr. Joel said, sounds like the question is when more so than how. All right, so one thing about genetic entropy 2.0, the original genetic entropy was used by a lot of creationists to argue for the youth of humanity in the earth. I've since softened that. I think there are better avenues to do that, but still, this is an argument against Darwinian evolution. So let's just go forward here. Genetic Entropy, that was published in 2005. And the title of 2.0, Destiny of the Human Race. Let's see. There's Dr. Sanford. He was the inventor of the gene gun on the left. That gene gun is in the Smithsonian National Museum of American History because billions of starving people were fed through his methods of gene improvement. He began the gene gun. Well, he began his career as a research scientist at Cornell while he was an atheist. He eventually became a Christian. He and his wife decided to go to church just to improve their marriage, and that's public knowledge. That's nothing too private. And in the process of going back to church, he became a Christian. He was a theistic evolutionist, then became an old earth creationist, and then a young earth creationist. So he was an accomplished genetic engineer, respected in the secular world, and now respected in the creationist world. So that's just a little bit about Dr. Sanford. There are a rising number of very elite secular scientists like James Loritan and Rob Stadler. Both of them are Harvard trained professors or scientists. And then we have someone Ivy League and John Sanford. So the face of young earth creationism is a little different than what you had 40 years ago. There are more and more of these. And that's why I'm just thrilled to see Peter here. And by the way, I'm sorry to just have to mention this, but the old internet followers of some of the popularizers, they kind of conflict with people like Peter and myself. And that's all I need to say about that. Some of the stuff of genetic entropy made it to the Journal of Mathematical Biology. This paper by William Basener and John Sanford broke the record by far for the number of downloads of any article. This is a secular journal, and let me see if I can show something. That led to something else, which I see if I could show it here. I think the editor, so that was a Springer Nature publication, and the editor probably noticed, he said, well, we're trying to write a reference book. We ought to, let's about mathematics. And well, they went to this journal, the Journal of Mathematical Biology, and said, well, look at the #1 paper. So they contacted Bill Baster and John Sanford and say, write whatever you want for our reference book, the Handbook of Mathematics and the Arts and Sciences. So that was a miracle of God that we ended up in these 4 authors, and I was privileged to be listed as the second author. Here, if I could make it a little bigger. So this is now on university library shelves and in secular universities. It retails, the book and the electronic version retails for $1,500. Now, I don't get a dime for it. I know a lot of you guys are generous and want to contribute to the channel. You can directly donate to me. Don't buy the book to try to get me some money. So I'm just telling you that. And so some of the stuff that we'll do here today will relate to those publications. And there's a lot left on the cutting room floor, so to speak. We had like 140 pages. We had to cut it down to 70. So there's a lot of material that didn't end up in the presentation today. So any comments or questions so far?
Yeah, Sal, I was just wondering exactly how widespread kind of, the idea of genetic entropy is among kind of, the secular kind of scientific literature. Because I was actually reading a paper on Neanderthals the other day and I saw that crop up.
It is very widespread. And since I know you're very skeptical and a research person, and I encourage anyone who's like, I consider kind of like, with a curious mind, and this has always been very satisfying. You can verify it for yourself, but I'll give you some names to start. Let me give you one. This is a book, Adam's Curse, by Oxford geneticist Brian Sykes. And he talks about how men are going to disappear because our Y chromosome is deteriorating. And I'm going to show you a graph that you'll see. It'll look hauntingly familiar to one you'll see in John Sanford's book. That is the decline of fitness over many generations. He is not unique in this. There is Gerald Crabtree, and we can have a little bit more detailed. Gerald Crabtree, there is Michael Lynch, Michael Woodley, and then there are other things like there is a paper on intelligence and childlessness, where intelligent women, they have a very high proportion, high IQ women have a very high proportion of childlessness. So that's an example of natural selection actually selecting for us getting dumber and dumber. And so genetic entropy basically means there's going to be more and more inherited birth defects across the population. We're going to get weaker, we're going to have more diseases to deal with, and so forth. Michael Lynch is another one. He's a very respected evolutionary biologist. I don't know of a single person of any great note that is tracking heritable diseases that thinks our genome is improving. And you probably, just to kind of reinforce that, you probably heard about the eugenics movement. Or you could research it throughout history where they're trying to do, basically prevent certain people from reproducing. But it was kind of that, that is something that reflects kind of the attitude that are, there's bad. They were either trying to accelerate the process of natural selection, or they were concerned about the deterioration. I mean, historically, maybe that's maybe a moot point in terms of our understanding today. But this would be an interesting research project to find out if you could find one geneticist that thinks our prospects as a human race are actually improving in terms of the genome. Now, one thing that's definitely compensating for that is our medical technology is growing by leaps and bounds. So some of these young individuals that have type 1 diabetes, juvenile diabetes, they have a chance of reproducing. So I had a dear friend, he'd be dead right now without modern technology, but he had children. And, that's kind of like, well, praise God for him as an individual and praise God for his family. But from a genetic standpoint, his children would be at risk of inheriting whatever caused them to have type 1 diabetes. So, so I'd invite you, but I would say the secular world is generally sympathetic. And emblematic of that is the fact Dr. Sanford got invited and received a warm reception by some quarters of the NIH. The people that were just hated creationists were really hostile. They're really upset that he got that invitation, but he spoke at the Mazur Auditorium, which is where they had the dignitaries like the presidents and Bill Gates or whatever. They rolled out the red carpet for him, and he gave his case. And because the NIH studies heritable diseases, there were people that are like, yeah, we see this. It may not, you can obviously see the problem for evolutionary theory. If the human genome with each generation is decaying, how then can we explain how we emerged, from simpler life forms, particularly intelligence? So that was a long answer to your question, but I wanted to give you some names to look up.
Yes, thank you.
So when some people, let me just give you a pointer. I have the various versions of genetic entropy. There are actually 5 versions so far, but they were never marked. So you get the re-release date, but the original one had the one with the flying saucer. That was the 2005 version. What I would encourage you to do, and the evolutionary biologists are very clever. They combed through this. They found some choices of words that I said, well, yeah, I think like Dr. Dan, I felt he had good points. And I said, well, maybe Dr. Sanford, I wish he didn't frame things in that particular way. One thing that Dr. Sanford got into was genetic entropy of viruses. And depending on what book and version you read, sometimes he's more forthright about viruses, and other times he wasn't in his original book. And I think, so just as a pointer, I would say the focus on human evolution. It's difficult to just go to you might find some organism that every organism and the way it reproduces in its genome is different. But if you focus on human evolution, I think and I know you love human evolution, I think you'll have a stronger case. So I felt Dr. Dan had because he's a virologist, he was I pulled Dr. Sanford aside. I said, I said, I don't understand the virus thing. I can't help you. There'll be a virologist going after you for that. And Dr. Sanford and Rob Carter published in that. And so the opponents of the genetic entropy hypothesis will just pounce on it. They'll just keep hammering the viruses. They don't want to talk about human evolution. So I just needed to advise you on that. So that's why I also came up with Genetic Entropy 2.0, which really focuses on the human. All right, so it's like, okay, so, we're talking about the viruses elsewhere, but I really want to focus on humans because humans are special. And I think a lot of what we have looks miraculous. And that's what I wanted to focus on. So I just, so that's some pointers. What I would encourage you to do is to look at humans and just work on that. Come up with Genetic Entropy 3.0.
Now, before I don't want to get us too far off track, but I understand that Dr. Sanford made a correlation between kind of declining age and, genetic entropy, particularly in the Genesis account following the flood. Is that correct?
That is
From what
correct.
I
And let me see
understand.
if I could find it.
So I was kind of interested in that in kind of 2 ways. One was, oh yeah, there's the graph.
Now,
Yeah, so
I want you to look at the
things,
shape. Did you see the shape of that graph? Okay.
Yeah, certainly exponential.
See, you're very smart. It's a decaying exponential. And I want you to see the graph in Dr. Sykes's book. Okay. Now, that's over a lot more generations, like 7,500. You may not see the number 7,500. But the general shape, yeah, that's 7,500 generations. So Dr. Sanford's is definitely more accelerated. And this isn't age, this is fitness. But
Right.
I want to show that there's a recurring theme of genetic deterioration, both in the secular world and Dr. Sanford's.
So do you think that, these declining ages could possibly be correlated to this? genetic
Yes,
entropy?
absolutely.
Kind
I'll
of in
tell
your
you
view?
why.
Okay. So you would
Yes.
agree with Sanford on that. Okay.
Yes. Well, for one, we have found these immortal cell lines in humans. You can look this up, and the viewers can look this up. There's the Gila cell line. Just look it up. What had happened, it really is a very sad story. Interesting scientifically, tragic on a personal level. Henrietta Lacks, and hence we named it after her, Gila. She had ovarian cancer. She died, and they were doing biopsies on her ovaries. They found that the cancer cells there, they were immortal. Okay, so cellular biologists know what the meaning of immortal is. It'll just keep replicating. It doesn't age. It may deteriorate more, but it's very important to medical research that they can have a stable cell line, at least stable enough. And it's like she died in 1953. 70 years later, these things are going. We expect them to go for thousands of years. And it's like, why does this keep going? We also have the Hydra, which is a eukaryotic organism, and it doesn't age. And in evolutionary biology, if you look this up, there's a problem of aging. They said, why do creatures age? We do have like the Hydra that seems to be a big counterexample to the rest of the animal kingdom. But it's like, why do creatures age? And so it seems that something switched off the ability to live forever. It seems like we, superficially say that we had the capacity in our genes to live a very long time. So I think that we can't absolutely prove it, but we have all these hints. And it does look, when we've examined, like, oh, this will be right down your field, Peter. There are bones that we've examined from 3,000 years ago. Okay, so this is, they're accepted like they would be in cemeteries or whatever. They would be accepted as really human remains, not some other kind of ape or whatever. The bone structure was the bones were thicker, the cranial capacity was bigger, so we were smarter. They said that the bone structure is consistent with also having very strong muscles. And I saw an article that said the average farmer would be like an elite CrossFit athlete. They were so strong. They were smarter and stronger. And there have been papers to that effect. So this is great that I'm having this
conversation. Because we do know that recently among Homo sapiens, there's been a downward trend in terms of cranial capacity. So generally, it appears that cranial capacity rose and then fell recently in modern times, which is interesting.
And then also look at the bone density, the strength of the bones.
Because we could certainly say that at least in certain individuals was higher in the past, because of course Neanderthals are known for having very, very strong bones, and some other different human types as well. One question about the aging. Go ahead.
Oh, go ahead. No, I mean, this would be a great research project for you to see if we've been declining in ability, and also there are historical records that would suggest we're also healthier. And I don't know how to establish that. But I will tell you, genetic entropy has a testable hypothesis, by the way. It's really kind of sad, because we're able to sequence genomes and track heritable diseases. 10 generations from now, we should know if the human genome is decaying. In fact, Michael Lynch said within 200 years, we'll have irreversible damage unless we do something now. Of course, unfortunately, it sounded very eugenic-like. All right. So this is a testable hypothesis going forward. You can test it kind of going backward, but maybe because it's vast, it might be a little sketchy. But genetic entropy is a scientifically testable hypothesis for humans.
So going back to that aging, how much So I've heard explanations of aging that have more to do with the telomeres, right? The repetitive strands on the ends of the chromosomes as being an important factor in aging, because they shorten apparently with age from cell replication. Is that something that would be connected to your idea of kind of slowing ages? Or,
This is very interesting because I was at the ENCODE Users Conference sponsored by the NIH in 2015. I happened to be sitting beside someone who studied telomeres for 20 years. And the short answer is telomeres are related, but it's probably a very minor part of the story, because he said the mouse have their telomeres are like they're immortalized, so to speak. They have the right kind of telomerase system that maintains the length, and yet the mice age. So that was a very important part. So correlation does not mean causation. You probably, just to emphasize this, reading ability could be correlated with shoe size. So as little kids grow, their shoe size gets bigger, but so does their reading ability. It doesn't mean that, okay, the size of their feet is the cause of their increased reading ability. And so there could be more going on, is what I wanted to suggest. It's not just the telomeres. We do think, now it's very interesting, if you actually go to the Wikipedia entry on aging, they will say it's still not well understood what causes aging. We do see a correlation with the degradation of the DNA in the somatic cell lines. As it replicates, there are 3 mutations per replication, and that does seem to do something. And that will contribute to aging, but we still don't know all the mechanics of what causes aging. And we do know one thing, that damage through ultraviolet light will increase signs of aging on the skin cells. You look a little older. So people that are in the sun, they get a lot more wrinkly and all. I've assiduously avoided sun, and I'll be 60 years old next year. And but I said early on in my life, maybe 30 years ago, I said, I'm just going to stay out of the sun. Now it's going to kill my vitamin D levels, so I have to take supplements now. But I said, I don't want to, because I saw this dermatologist. She was like 50 years old, and I said, oh my goodness, she looks like she's 25. I said, I want to be like her. Okay, so that's a little bit of vanity on my part. So that is an example of DNA damage. Because she was saying, oh, she saw these girls out there sunbathing. She said, oh, they're going to be my future patients. She's a dermatologist. So that would suggest there that DNA damage does accelerate aging, at least for skin cells. So, all right.
I'll let you get back to your presentation. Sorry for taking you on that whole tangent.
No, this is okay, Peter. I'm here at your service, and I think because I see that you're an aspiring researcher and have a curious mind. I think you're going to be very successful coming up with your own version of genetic entry. So there might, and that's happened with me and Dr. Dan. Covert cuttlefish. He said something really nice. He said, Sal, you look great. I was a lifeguard for years, and my cheeks from the glare and from the sunglasses and pools have sun damage. Wear your sunscreen. Okay. So a little advice.
Wear your sunscreen. I did not today, which is why I look red.
So I'm just I'd like to share, this is tonight for you. And I really like your approach to science. When you hear the other side do criticisms to my ideas or anyone else's ideas, and you think they're right, I take no offense. I'd be grateful if you find out something that is wrong, and you don't let it propagate, and that's fine. So, Dr. Sanford didn't hire me to be a yes man to everything he said. Now that caused a little bit of friction, when I said, Doc, this isn't exactly the way. And that even goes right down to the title. Entropy is from second law thermodynamics. And there are a lot of creationists that think the second law of thermodynamics is a good argument for creationism. And well, I don't think so. All right. So I would say use entropy as a figurative term. It's okay. If you try to apply the second law of thermodynamics to genetics, I think you're going to get in trouble. So that was kind of our first point. And I said, Doc, I did, if you have another edition of this, I think you need to rewrite 2 or 3 pages. I'll help you rewrite it. Okay. So, I'm here at your service. If you find something that you think you can improve on, I really encourage you to do that, because this the basic idea is now very defensible. And some things, just because we had to correct something doesn't mean things out of the, you're going to throw the whole idea out. I'll give you one small example. In the first version, Dr. Sanford said, all mutations are deleterious. There are none that are beneficial. In the later editions, he softened that. He said most. It got corrected, and it's going to throw out the whole theory because there were some minor corrections, but the major hypothesis is supported. So this is great. I was actually hoping you would ask me questions, because then you'd bail me out of having to do a boring lecture. So Rebecca, you've been silent. Do you have any thoughts?
Well, there was something came to my mind when I interviewed Dr. Dan on my channel. I kind of asked him about mutations, like, well, why don't we marry our brother and sister? If mutations are good, then, isn't the whole reason that we don't, do inbreeding like that is because of the mutations that collect, right? And he said, like, no, he said, no, basically, like, it's a separate thing. And, because there's a few bad mutations that we're trying to avoid. There's like, a couple bad mutations that if you're, genetically related, then there's a higher chance of getting them. But in general, like, I don't know. What are your thoughts on that?
This touches on what they call killer recessives, and it's very complicated. But the idea, so if you're not marrying your siblings, if you have bad genes that they call recessive genes, where the mother to have that particular trait, the proper term is allele. If they both have it, then it'll express itself as bad. But if you marry someone else who doesn't have it, there's a good chance your child will not express that trait. It's a little bit complex. It goes into Mendel's laws of genetics. There are 3 of them. One of them is called the law of dominance. But let me just point this out. You can find literature that will say most mutations are deleterious. Most mutations are deleterious. It's for the simple reason that it's easier to break something than to make something. Easier to break. Now, how we arrive at that has been, Dr. Dan and I had a debate, and he was contesting how Dr. Sanford interpreted a particular work. I would argue Dr. Sanford and not with Dr. Dan at this point, but it's all moot because there's so much literature that say changes are usually bad, and so it might have flown just 20 years ago. It's starting to be compromised, and even in the last things that they presumed were neutral, what they call synonymous mutations, they're finding out they're mostly detrimental. And then added to that, anything that is considered beneficial in darkness, that means it increases reproductive success, it turns out those are usually damaging the genome, or could be. So it's really tough now to say that that things aren't breaking down. And I'll point out that the research that I was hired and I got to work on was on proteins. You'll see proteins connect very precisely, just like a car. And so the reason that a lot of mutations are damaging is you can't just change the shape of something and expect it to keep working with the things it's connected to. It's that simple. I mean, it's more complicated than that, but I wanted to, that's why it's so hard to make a truly major change in a protein. And I've cited things like the potassium ion channel. It's like, it's precise within angstroms. If you make it a little smaller or bigger, it's done. You're not any better by mutating it. You would hope that when you throw changes at it, that at best it doesn't damage it too bad. And you'll see this principle over and over again, where anything, maybe an isolated protein that is an enzyme, you might be able to increase its enzymatic activity. For eukaryotic cells, eukaryotic creatures like us, where there's so many interconnections, just like parts of a car, you start shaping things a little different, it's going to break down. And that's what I've really focused on the last 8 years of my research, was trying to show, okay, let's see if we can kind of show intuitively why mutations are just generally bad. So anything with connections and geometric fits, very easy to break. Very, very hard to make a new, if you're going to make something bigger that connects to something else, you have to simultaneously make both of them change shape. And look who is here.
I apologize.
Emery.
Hi, Emery.
Hello, Emery.
Good evening, all. I apologize. I did tell Sal in advance I'd be late, but I did not expect to be 45 minutes late. I apologize.
No problem.
That's right. This show, I don't know if I mentioned it. The show is really oriented to Peter here, so he's the special guest, and he's just been leading the kind of the questions that I'm trying to answer, and that's because, Emery, if you and I started talking, you know where it's going to be. We're going to just blow away our audience with nerd talk. You remember that whole R.H. Brady discussion we had?
Yeah, I haven't forgotten R.H. Brady.
That went on for 10 hours, Peter. And you talk, and we were trying to get Rebecca her stripes in a nerd torture session. She lasted an hour and 15 minutes. So that'll happen. In other words, don't sweat it. It was just mostly.
That sounds like competition even for Rebecca's streams.
It wasn't all at the same time.
My streams are fun. They're not nerd torture.
And I will also point out that my stream with Sal was over the course of a week, not all at once.
Okay, I'm talking on my secondary computer. Can you hear me?
Yes, it's much better.
That's much better, but there's now an echo.
Yeah, he has to mute himself.
Okay, Dr. Joel, thank you so much for the feedback. So you audio check again. Can you guys hear me?
Yeah, it sounds good.
Yes, it sounds a lot better.
Okay.
Yep.
All right, guys. Sorry about that. So now you can't see me, but you can hear me better, which is more important. Let's see. It's going to be funny going between two computers.
Well, while Sal is working on that, I'm going to go ahead and say good night. I'm going to still be listening in on you guys, but I told Sal I was going to have to leave in an hour. And since we're paused right now waiting for him, I think it's a good time to say good night. So thanks, guys. See you
thank
later.
you so much, Rebecca.
See you,
Lisa,
Rebecca.
Great moral
Bye.
support. God bless you.
God
Have a
bless
good
you
night.
too.
All right. Peter, in case this ends up being too late, we can always continue. But I wanted to at least get you on a starting footing. And so, Emery, just to catch you up, I was telling Peter that there is the original book by Dr. Sanford, and the evolutionary critics will go after his stuff on viruses. And I was just saying, I was encouraging Peter just to focus on the human evolution thing, especially since that's his specialty anyway. And I'm at the point, it's like we could, for the sake of argument, say genetic entropy doesn't apply to viruses, or even if it didn't, it can't be generalized to say it doesn't happen in humans, because we have so much empirical evidence that our genomes are deteriorating. And so I was encouraging Peter to pursue this. So I'm laying this out also. I told Dr. Dan that we'll have a separate night. He could, reflect on what I said tonight, because Genetic Entropy 2.0 is kind of my version of genetic entropy. But tonight is Peter's night to ask me questions about it. So anyway, let me see if I could find. All right, so I'll show you, I'll illustrate something with kind of the principle of all. This also explains the origin of junk DNA. So you were asking about what do the secular people feel. So for human evolution, Dr. Sanford's definitely in agreement with the mainstream. So Dr. Sanford addressed the National Institutes of Health, October 18, 2018. There's Michael Lynch. This is Mutation and Human Exceptionalism, Our Future Genetic Load. And he's arguing it remains difficult to escape the conclusion that numerous physical and psychological attributes are likely to slowly deteriorate in technologically advanced societies. The incidences of a variety of afflictions include autism, male infertility, asthma, immune system disorders, diabetes, already exhibit increases exceeding the expected rate. The observational work may substantially underestimate the mutational vulnerability of the world's most complex organ, the human brain. Because human brain function is governed by the expression of thousands of genes, the germline mutation rate to psychological disorders may be unusually high. At least 30% of individuals with autism spectrum disorders appear to acquire such behaviors by de novo mutation. It has been suggested that there has been a slow decline in intelligence in the United States and the United Kingdom over the past century. And some will say, well, that's because we don't have, we have modern medicine, or we're not eliminating. But I just wanted to throw that data point out there, because where maybe geneticists like Lynch and Sanford would disagree. Sanford would say, well, certainly if we had more higher reproductive excess rate and we didn't have modern medicine, that would slow genetic deterioration, but it's still there. So that's just an example. There's, translation, we're getting sicker and dumber. So how can Darwinian evolution fail if there's survival of the fittest? So in one scenario, the way Darwin envisioned it, this hypothetical example here. Let's say we started off, we had a population where 20% of the population acquires new mutations or slight birth defects in each generation. So, let's apply kind of Darwinian selection here. So let's also assume we have perfect selection, and we eliminate the 20% of the population that are mutants. That's not exactly true, but let's just make this very ideal. And you'll see there's the Terminator here. He's going to eliminate the, the, the defective individual. And so the survivors become parents of the next generation. They have a clean genome. So so far this looks good for Darwinian evolution. But what about, suppose all of them are mutants? Suppose all the children of the previous generation have at least one small birth defect. What can we do? Well, we certainly can't wipe them all out, because that would wipe out the population. So maybe we'll just get the worst of them, and the next generation will be not quite as good as the previous generation. But this is still survival of the fittest. But you could see that there is kind of a semantic issue here. The fittest genome has actually decayed because it was the parents. So survival of the fittest is really fittest among the siblings or the peers. But what Darwin failed to account for is, what if the fittest genome was the parents or the ancestors? And that's the difference. I'll pause here if you have any thoughts, Peter.
Yeah, so in terms of kind of fitness, I mean, how do we get a population like you're showing here where, we have these effects which completely, destroy like the fitness of an individual? Because, I mean, generally that would seem to be rather rare, right? So creatures that would get a mutation that would, completely destroy or, severely damage their fitness wouldn't make up a lot of the population. So how exactly could we kind of get to that point?
All right, I may not completely understand your question, and I apologize. Let me just try to say this. Where genetic entropy is particularly bad is if the mutation isn't severe. Like,
so that in that case, it can't be selected out, you're saying, and thus they will accumulate.
Yes. So let's just say we're getting rid of the most severe. And there is a problem with the definition of fitness. The evolutionary definition of fitness is more babies. So if you look at Joe Felsenstein's graduate level book on population genetics, it basically says you take the number of babies that are made by an individual really associated with a genotype or an allele. They like these one locus models, really one gene models, because the math is easier. But let's go to the just one locus haploid model, and that's easier mathematically. You could see fundamentally the definition of fitness is reproductive abundance. So the number of children, first it's the number of children, and then you go by what they call viability, since not all of them are expected to reproduce. So you take the number of children, you multiply it by some percentage called the viability, and you end up with the number of offspring that go on to reproduce. That's the evolutionary definition of fitness. And what had happened, I think the most embarrassing thing that happened is, and I'm going to, let me share my screen here. The most embarrassing thing that happened, you could see it here. Let me, in my essay, Genomes Decaying, but Darwin Always Gaining. So this is from a peer-reviewed paper by Lenski. Have you heard about the Lenski's long-term evolution experiment? You'll hear that.
Yeah.
All right. One of the papers associated with that, Lenski was a co-author. The lead author was Kochi. In the title says, Genomes Decay Despite Fitness Gains. I want you to chew on that. Genomes decay despite fitness gains. If I had to say anything about a major problem in evolutionary biology, it's that phrase right there. Genomes decay despite sustained fitness gains. What that's saying is that the reproductive abundance, which is Darwinian fitness, increased, but the genomes were losing genes. This is a very bad definition of fitness, in other words, that's adopted in evolutionary biology. So Dr. Sanford's original book, Genetic Entropy 1, what I call 1.0, it used the evolutionary definition of fitness, which is reproductive success. Our later writings began to say, well, you know, that was generous on our part to assume that, but in reality, this is genetic deterioration. And the way to look at this is like, they use the word loss of versatility. So just imagine a hiker, who has a lot of equipment that will enable him to survive in a lot of environments. So he might have extra coats on a warm day than he needs, you know, extra blankets, etc. There'd be another hiker who has a lot less equipment, and he'll just be moving and hiking along well in one particular environment. But let's say he has a really cold night or adverse weather conditions, he's toast. And so the loss of versatility is losing capabilities that would enable him to thrive in other environments. And that's what the experiments were revealing. And so how do you account for that? You could say, well, he got better in this environment, but he lost this ability in other environments. So you'll see creatures like wingless beetles or wingless creatures, or all these creatures that lost organs, and you're like, well, yeah, the fitness gained, but it just trashed the genome. It lost versatility.
Now, this kind of strikes my interest, particularly because of, you know, diversification, right? How we get creatures to diversify and speciate and all of that. Do you think that genetic entropy kind of provides a barrier to that? Diversification, meaning that rather than appealing to kind of the more usual kind of, you know, mechanisms of natural selection and mutations to occur for diversity, that we have to look more primarily to created heterozygosity or other ideas like that?
Okay, this is outside of my area, and we have Emory here. I would say, and then we have Randy Galuza, the president of ICR, the Continuous Environmental Tracking. I think first off, there's a lot about the genome that we presumed that we don't know. But as far as when we see kind of what we call speciation or whatever, or rather than speciation, why don't I call it specialization?
Sure.
It generally, to me, it looks like it's a loss of heterozygosity. That's what it looks like.
It's called the founder effect in speciation literature.
Am I off the wall here, Emory?
No, you're not. You're not. Now, it depends on how the species forms, because if you're getting two existing species hybridizing together, you can increase heterozygosity, because you're essentially mixing things that haven't been mixed in, you know, 100 years, 200 years, however long it's been since they split. So you can increase diversity depending on the split. It really depends on how the species is forming. Like the new lineage of Galapagos Island finches, Darwin's finches that formed during the Grant study there. I don't think they have the numbers. They might. I don't remember for sure. It's been a while since I read the book. That one might be more genetically diverse than its parent species, just because you're mixing two things that hadn't mixed in a while. But by the same token, if you had, you know, a founder effect take place where you had a population get split in two, you're probably losing diversity from both populations. So it does depend somewhat. The other assumption here that doesn't get talked about very much is, and this is actually based on a paper that just came out, which I read actually yesterday because I was reading it for some other purpose. But it turns out that it's not just non-synonymous mutations that decrease fitness. It's synonymous mutations too. And they decrease fitness, the evolutionary version of fitness, by about the same amount, like within like 0.1 or 0.2%. So essentially what we're looking at here is that there's very, very skimpy evidence for truly beneficial mutations. And by truly beneficial mutations, I mean mutations that are not mutations that increase evolutionary fitness, because that can happen. But mutations that will actually, I want to phrase this correctly, mutations that won't break something in the process of conferring a new function or increasing fitness or what evolutionary version of fitness or what have you. And the paper I was looking at, it was like, even them, they admitted that the number of mutations that they looked at, and it was like thousands of mutations that they looked at, was like 1% they considered evolutionarily beneficial, which is peanuts, considering they looked at like 8,000 mutations or something like that. And the vast majority are deleterious, or they're negative for the evolutionary version of fitness. Now, what that means is that to increase diversity in a species, to get the level of diversity that we see in the present, there's not enough time. There's just mutations ad nauseum missing from the record. You can't go from 4.3 billion years ago, we get the first living cell with probably, even if I give them a billion base pair cell, which is unlikely because most bacteria are only a few million base pairs. But even if I give them a billion base pairs, to get to the human genome, which is what? Oh, good heavens, Sal, how many base pairs in the human genome? I'm blanking.
About 3.3 gigabases. Yeah,
3.3 gigabases. Yes, thank you. In 4.3 billion years, you got to do a lot of mutating, and not only that, you got to get them all to stick, and you got to increase the genome size. It's mutation. I think there is potential for mutations to be somewhat increasing of diversity. I'm not throwing it out completely, but I mean, to me, at least from the models I've read, and I haven't read everything, obviously, but what I've read, I think the blanking on this too. I'm tired. CHNP model is probably the most effective model that we have at the moment. So that's my 2 cents. Take it for what it's worth.
Yeah. So I just say, I think there's a lot we don't know, Peter, and I'm sorry I couldn't answer your question. And so you're the next generation that might explore some of this for us.
Yeah, I'll back Sal up on that. There is a ton of stuff out there that we don't know. I just read an article that was actually by Dr. Galuza and a couple of co-authors in CRSQ, where they're talking about how the Mexican cavefish goes from blind to sighted and then back to back and forth, essentially within a couple of generations. I think there's some very interesting stuff going on there too. So it may not all be genetic. There may be some epigenetic ability to make some changes. I'm skeptical because of the example of Darwin's finches. Epigenetics tends to only stick if the environment is constant, and the environment is not necessarily constant. And after the flood, it would not have been very constant at all. So I'm a little skeptical, but I think that there's at least some potential there for something that could also play into the diversity. So again, as Sal said, a lot we don't know, a lot of stuff still up in the air.
So going back to exactly, how we can generate the diversity that we see, a lot of people would obviously appeal to, created heterozygosity. Now, for me, I'm not sure whether that can explain a lot of the diversity that we have, simply because, there's too much variation within the human genome for them to have been that diversity to have been represented in Adam and Eve. And often then I'll hear, well, I have before at least heard people appeal to Adam and Eve having, reproductive cells that would have had differing information from their own cells, which to me seems kind of somewhat ad hoc. But do you think there's a case to be made then, for created heterozygosity playing a pretty big role in terms of diversification?
I would say yes. If we just, you can actually see it when you study Mendel's laws of inheritance. Even with just a few alleles with one just one gene, you can have a great variety. That was with the peas. And you just look then at, say, 10,000 loci or 20,000 loci. The way you can, the phenotypic diversity could be tremendous. So according to Mendel's laws of inheritance, because of the law of dominance, the 3 laws, I only know that one. And I'm not a genetics guy. I'm a protein guy. But even with that, you could see then what ends up happening with the phenotypes. It's which ones you end up being dominant, or you have homozygosity in a position. And the important thing, and this is where I credit Nathaniel Jeanson, he said, we have known examples in horse breeding. We know who the founding population was, and it's just astonishing all the phenotypic diversity you can get, and then you can trace it in terms of the the mix of genes. It is kind of shocking. So that's why also to me saying diversity, that we look at phenotypic diversity, and I'm just like, it's not as precise for me that it's just like to focus on just one protein at a time. You can definitely see it when you're looking at just one gene. It's like, well, no, you're actually losing diversity over time, unless you do mutations.
So I'm gonna put in just a little bit here, if you don't mind, Sal. So I'm open to the possibility that mutations can create diversity, but it depends on what you're doing by diversity. If you mean a gene that creates red hair, if you break a hair color gene and you create red hair that way, okay, you've changed the hair color, and certain places in the world, red hair is really not a positive beneficial trait to have. But if you're living in Norway, where the sun only shines for what a month a year, it really doesn't matter that much. So it may not do very much harm. So again, I'm open to the possibility of mutations being involved, but I think most of the heterozygosity we see in the genome is created, or it comes about through regulatory gene processes, because we know now that a lot of genes are under regulatory control from other genes. And so you switch one on, you get a different phenotype. You switch it off, you get a different one. I think there's, again, I'm going to echo Sal. I think there's a lot we don't know, but I think I like the trend. I think we're on the right trend, but I think I think that there's, again, a lot more work that needs to be done, because any geneticist will tell you this. They're great at generating millions and millions and millions of pounds of data, not so great at actually sorting through the data. So those of us who are your age, Peter, are the ones who are going to have to sort through the reams and reams and reams of genetic data to try and make sense of it.
Okay, here's a good example. Emery, this is a Punnett square. Okay, I'm going to embarrass myself. You got to help me here, buddy.
Okay.
So let's say we started off with yellow circly peas. So this is circly pea, and Mendel's laws of dominance. We have R means round and Y means yellow. We need to find one that is R and something with green. Do you know where I'm headed with this? I want to see if I can start with a P that has everything, and we're going to get all the diversity. So,
yeah. I mean, just by starting with a heterozygous round yellow for both traits, you can get green peas, you can get wrinkled green peas, you can get non-wrinkled green peas.
So the issue there is really in the...
Go ahead, Peter.
Sorry, go ahead.
I was about to say, would we start with this? I'm sorry, Peter, go ahead. Go ahead.
So from what I've heard of critics, right, their argument is that basically there wouldn't have been enough, basically there's not enough diversity in the loci, right, because Adam and Eve would have only had, these specific loci. and thus they could only have so much. And since we see more diversity in those loci than would have been in Adam and Eve, that at least some of that had to come from mutations.
Again, I don't have a problem necessarily with that. And I will say that I stay out of human evolution and human genetics because I just don't enjoy it. It's not fun. But I don't necessarily have a problem with some mutation being involved in creating certain diversity. And a mutation by definition does create diversity. So not all diversity is good, right? Because you're introducing I don't want to say allele, a nucleotide that was not there previously in the population. So you are creating some form of diversity. Now, the problem is most mutations have a negative effect on fitness. The question's not so much, and that's evolutionary fitness, not the version of fitness that Sal's talking about. So the question is not so much, did Adam and Eve have enough loci to cover the entire diversity of humanity? The question is more, I would think more along the lines of what did they have that did they have enough to explain the majority of it? Okay. Sorry, I'm reading the chat. Did they have enough to explain the majority of it? And then we can, I don't mind looking at the non-typical cases and saying, okay, what's the origin of this? Where did it come from? And does it act? What does it do? How did it get there? Is it something broken? Is it something, that doesn't work anymore? Did it break something to cause that? What's going on? So I think you're going to need, again, a lot of stuff we don't know here. Would require a lot more work than I have had time to put in, and I know other people have put some time into it, but not as much as probably we need to. But we are limited on people.
I will say this, like Dr. Dan here, Creation Myths, he comes up with some figures. I've told him I think that's a good objection to the creationist model, and I don't have answers for it. That's why we need new researchers to help tackle some of these problems. And so I congratulated him on actually finding some of this stuff, and just like, okay, you got me stumped on that one. So the old Earth creationists would probably be thrilled with that. The young Earth creationists have a problem. And for me, that's why I like to start with old Earth creationism first, because it's a little bit more defensible from our present knowledge base, not from a biblical standpoint, but I'm just saying, it's still creationism, but, we're not under the gun in terms of a time constraint. But I wanted to go back to this diagram here. Correct me if I'm wrong. And we got a biologist here like Dr. Dan who teaches biology, so he'll ding me if I get this wrong. So presumably we could just start with a P that has this configuration, the capital R, lowercase r, and capital Y, lowercase y. It's phenotypically, it's a round yellow P. But then future generations, you could have 4 different phenotypes here. And you can also imagine that natural selection could generate this. So let's say you have 20,000 loci. I can't do the numbers off the top of my head, but that's a lot of diversity. And Nathaniel Jeanson was trying to point that out. He said, look at the number of combinations you could have with this. And that's just in terms of the heterozygosity. And as Emery pointed out, there are probably other things that can even modulate it even more if we include the effect of non-coding DNA.
Mm-hmm. Yeah.
So you would basically argue that given we have enough heterozygosity, it doesn't really impede, speciation or, adaptation to new environments, even with the effect of genetic entropy.
Okay, so
at least soon after the flood.
Oh, can you repeat that again?
So I'm saying basically, genetic entropy could possibly, be a barrier to, speciation, right? If enough accumulates, populations become very homozygous, right? But you are saying that given enough created heterozygosity, there's time for creatures to diversify before they become bogged down basically by genetic entropy.
I think that's a fair assessment in general. I don't think genetic entropy has to be a barrier necessarily. I mean, because you can get speciation without any change in genetics. All you have to do is get a variance in mating behavior, depending on what definition of species you're using.
Yeah.
Which
Go
is
on.
a whole nother kettle of fish. And you send Dr. Dan down that rabbit hole sometime.
Okay, this is where I want to point something out. Even the evolutionary biologists, Dr. Dan gave I don't know if you were there when, it was Guzman who was with us. It was Ask Me Anything. It was Guzman, Emery, and myself, and we were asked, and maybe Rebecca. Dr. Dan had an Ask Me Anything. And I brought up, I said, speciation, Dr. Dan. And he said, well, we had a whole class, a graduate class on it. By the end of the class, no one could agree on the definition of species. The faculty and students were arguing amongst each other, and it was glorious. So here, get it from Dr. Dan here, right here.
Species are fake.
Okay.
Yeah.
So the reason I really, okay, let me just say here, I sympathize with that. It's better to just say different phenotypes, because those are, you can define a phenotype, right? You can even define the genotype. You can define the genotype and its associated phenotype. Then you don't get into these endless arguments as what a species is. We can be a lot more precise in the modern day now that we have genetics. We can at least go to the genetic level now. I mean, if we had enough sequencing power and we could sequence an entire population, we can just say, okay, whether you call it a species or not, we can identify genotypes. We don't have to just add this layer of confusion, which is the species. And Schnoil said, I don't like Dr. Jeanson's attempt on speciation. I have to agree here. When he made that presentation, and I love Dr. Jeanson, and here I are colleagues. By the way, I do need to say some people have been circulating ugly rumors that I said things about Dr. Jeanson that I absolutely did not say. I've been accused of calling him a hack or whatever. I did not say that, and that came from a certain community I don't need to mention.
Yeah, I know where that came from, Sal.
Yeah, that I did not say that. And if I see Dr. Jeanson, I'll say, please ignore those guys. Maybe don't ever show up on his channel or coddle them, because they're attacking people like me. They're attacking people like Peter.
Did you hear I'm getting whacked by Kent Hovind and
Can I join you in that department?
Oh boy! Ow!
I'm the geologic column, whether it exists or not. So that'll be fun.
Okay, I'm not joining you for that one, because the only rocks I know anything about are the ones in my head. But it's just funny how things change, because I grew up on him and I used to like him, but things change.
Okay, so Dr. Dan here has an objection. He said the loss of heterozygosity thing doesn't work, and anytime Dr. Dan puts something up there, it's like, I'm not going to just write it off. It's worth exploring. So, look at that.
And here we go. We got Dr. Dan versus Dr. Duff.
Okay. There you go.
I told you guys that topic, man.
Okay.
Anyway, sorry, I didn't mean to start World War III. I really didn't. I apologize.
But we had to bring it up because Peter was framing his question in terms of species, and
Yeah.
I just cringe because genotypes are very precise. Can we make new genotypes? And it's very let's say hypothetically that all we started off was this. Can we make new genotypes? Absolutely. You have 4 right here. So depending on what the founding configuration was, you can absolutely make new genotypes. That shouldn't be controversial. Okay, so here we go.
I would sit down and watch. That's one of the few debates I would sit down and watch. Is Dr. Dan versus Dr. Duff on whether a species is real or not.
Okay, so Peter, going on an hour and 20 minutes. I'm loving this conversation, and I could show you more things.
I'm going strong. Yep, let's go.
Alrighty. So I had kind of my prepared thing, and it's really great to have these side discussions because I want to be of service to you. And so let's go back to here. Going back to this, we're not going to. Can we just focus on genotypes rather than the question of what a species is? Because we won't. I don't think we'll have arguments.
No, I know. I was just trying to basically say the origin of kind of this new group that is somehow separate, basically, genetically,
Yeah, so
in terms of breeding.
I'm just going to go with genotypes. I'm sorry. Okay.
That's
and
good.
As the biologist, yeah, okay. We can be safe there. Anyway, and you have access to the book Trace. I haven't read it.
My copy is on the way right now.
Okay.
I have a copy that I haven't read yet either. I'm graduate school's taking first priority.
So you could see intuitively the problem. Now, this is a simplified haploid model, just not sexual reproduction, but it can be extrapolated to the diploid where you have mothers and fathers, but you have to have a little bit more mathematical sophistication. So what I'd like to point out is Dr. Grauer actually recognized this problem. Now, if you have sexual reproduction, you have a little bit of an out, meaning going back to that Punnett square. Let's go back to the Punnett square here, and you could see how this could kind of work. You could see here, if you have, you could have one individual that'll have a genotype that doesn't, that's all one class. So you can imagine then, if we've had all the children, the mothers and fathers having this mutation, it's possible they might come up with one genotype that is clean. And that mathematically it somewhat corresponds to this situation where you have this one genotype here. But you need to have for that to happen, you have to have a large population from which you can select the one clean genotype. That means that one P will have to make a lot of varieties of Ps. Now, with humans, that's going to be difficult, but this is a little bit comical. That so when you have males and females, you can get a little bit of an exception. And based on the rate of deleterious mutations per individual, per generation, there's someone named Dan Grauer who did a calculation on this, and it agrees very much with mine. And he was saying, okay, if most of the genome is functional, this is the outcome. And this is the infamous quote. He said, if ENCODE is right, then evolution is wrong. ENCODE claimed that most of the genome is functional, like 80%. And I've cautioned creationists. I said, don't cite the ENCODE study. Just wait around, and there'll be more studies. And I've covered that. You came to my stream recently that covered why I think the ENCODE consortium is right. So therefore, evolution is wrong. And I'll show how he arrived at that. So let's just look again. Let's just say we started off with one individual here that had 0 bad mutations, just hypothetically. Just hypothetically. And this is the haploid case. It can be extended under free recombination, et cetera, et cetera. There are all these qualifications here, and I'm assuming a Poisson distribution, and that's accepted in population genetics. We don't have to get bogged in the details. Just for the sake of completeness, I'm mentioning it. But let's say generation 0 has no bad nucleotides. And so some of the offspring might have more than others. But if the average number is about 80 here, you can have some with 100, some with 60. So Darwin and Spencer was the one who said survival of the fittest. Darwin came up with, he adopted survival of the fittest in the 6th edition of Origin of Species, but credit goes to Spencer for that. So let's say survival of the fittest. So let's, among this, just pick the best, or the best of the work, the best of the defective ones. And I'm just going to illustrate this. So the parent dies, rest in peace, and we eliminate the worst. Well, this one goes on to be the next generation, and this is with 100% efficiency of selection, which is very difficult to achieve. Because accidents can just wipe out someone who happened to be the best genotype, and he gets run over by a car or struck by lightning. Anyway, so now this is now generation 1. We're just going to repeat the process. We add an average of 80 bad mutations, and then we'll just select from the best of the remainder. This one dies, the parent dies, and you could see rinse and repeat. This is pretty much like the original diagram with Arnold Schwarzenegger. And you could see that, oh yeah, by, so we're going to fast forward to 1 million generations. I don't know why I'm not, oh. So we just fast forward 1 million generations. You see that this is irreversible. And, they have things like mutation. Let me just pause a little bit here. In my debate with Dr. Dan, he used the word mutation selection balance and all. If you adopt, but that's assuming that flawed definition of fitness in evolutionary biology. Once you adopt the kind of more conventional sense where, it's actually, you have genomic decay and loss of versatility, then that's out, mutation selection balance is kind of out the window. You can still have genomic decay, because I showed that quote from Lenski's experiment that the genome decays despite fitness gains. So you have to be, some people will say, well, it'll balance out, and there'll come a point you average out where you have, you get to a point where it doesn't get any worse in terms of beneficial and deleterious, but it depends on your definition of deleterious and beneficial. Okay, I just need to point that out, because if you adopt the more kind of common sense version of what it means to decay or lose versatility, then the distribution that I was showing here is, this is definitely in play. Okay, any questions so far?
I think I'm good.
Okay, so, but even under the evolutionary definition of fitness, that's where Kimura, Ono said the overall deleterious mutation rate per generation becomes 1.0. Now, he had a different notion of what we consider, he was going by genes rather than individual nucleotides. And we're finding, thanks to this paper the last month, that every nucleotide counts for a protein coding. That whole thing about synonymous and non-synonymous, everything in the coding region counts. It's not just the gene. But this is for historical purposes. This is what, 1972, 50 years ago. But he said the overall deleterious mutation rate becomes 1.0. This relates to the idea you can't have more than one bad mutation per generation per individual, given the reproductive rates of humans. And you have to go through the Poisson distribution. It's a little bit complicated. We can walk through that if you ever want to be tortured. It's like an hour-long discussion. But it kind of goes back to, all right, like let's say every child on average has at least one mutation that the parent didn't have. You'll get a situation where it's just going to accumulate. You can't reverse it. And I call that Muller's limit. So go ahead, Peter.
Could we go back to kind of your redefinition, or perhaps what you would call the more common sense definition of fitness? Now, listening to Dr. Dan, I listened to a video of his about genetic entropy recently, and he was trying to kind of put forth the argument that basically, as an organism's genome would decay, it would basically eventually result in, a lack of production of offspring, meaning that basically these two terms of uses of fitness basically result kind of in the same effect, which is less offspring. Would you kind of agree with how that?
No, not necessarily, because or at least in the short term. So and it's really, I'll tell you why. We've had some species of yeast that just keep going, and we know they've lost genes. There's also, you can search for it, something called the pangenome. And let me show you how many. Okay, it depends on the creature. You can't all right, let me see if I could show this. Picture's worth 1,000 words. I'm going to show the pangenome of E. coli. See if I can find it.
I hope I didn't misrepresent what Dr. Dan was claiming there, but that's what I thought his argument was.
Let's see.
Well, while Sal's looking for that, I don't think that it necessarily implies that just because you're building up genetic mutations that you're going to immediately at least start losing offspring number. It depends on where the mutations occur. The concept of genetic load is something that's well known in the conservation literature, and they talk about it where your organisms get less essentially loaded down with mutations, and they have to do what they call genetic rescue, where they'll introduce other organisms from outside the population or even sometimes outside the species to try and keep the species alive. So they're familiar with the concept. Now the genetic load is slightly different maybe than genetic entropy. They're defining it a little bit differently, but it is it's a similar idea. And it's something that they know about, and they know that it's there. They just don't. Unfortunately, sometimes these disciplines don't talk to each other, and you'll have people in the population genetics literature who will argue against things like genetic entropy or try to solve that particular problem, but then won't consult the conservation literature and look at where it talks about things like genetic load, genetic decay, even things called an extinction vortex. Now that you can get out of that, but generally it involves genetic rescue or something along those lines. So anyway, Sal's found what he wanted, so I'll stop there.
Okay, so I wanted to point out that biology is the science of exceptions. Meaning you try to make a general rule and you're going to find an exception. So it's easier if we do it on a species basis. So your question about will it lead to lowered reproduction or extinction? I would say, first of all, it depends on the creatures you're studying. For bacteria, it doesn't necessarily. Here's a good example for the viewers there and also for you, Peter. You could look at what we call the pangenome of E. coli. So we have bazillions of sequences of E. coli that seem to be thriving. And it turns out the core of E. coli is only about 20% common. So you have all these strains, and I didn't use the word species, I'll use the word strains of E. coli. And some of them have radically different sets of genes, but they have this 20% core. So that could suggest you could have an E. coli that has 20% of the size of your typical one, and it's still alive, and it may or may not be thriving. Now, do you think, I mean, just a rhetorical question, does anyone think we could lose 80% of human genes and still thrive? I would seriously doubt it. Because bacteria are special, they can have a lot of versatility that they lose, but humans are so specialized in what they do. I think if you lose even half of our genome, we'd be dead. We might be kind of like on life support, but we wouldn't be able to thrive. So I would say, however you interpreted what Dr. Dan said, I would say it really depends on the creature under investigation, the class. And then you would go from there. And so that's about all I could say about that. And
Sure.
so maybe I didn't answer your question very well, except to say it depends.
So how necessary kind of to your model do you think this new definition of fitness is?
I think it's very important.
Okay.
Okay, because I'll give you just maybe a simple explanation why. Absolute fitness is defined by just the sheer number of individuals. So in a sense, the human race in terms of absolute fitness is really just prospering. You have all these, the exponential growth rate. And, if the atomic bomb hits and wipes everyone out, then the fitness goes to zero. And I need to point out something else. That's the problem with the evolutionary definition of fitness. It's outcome based. So if there's an environmental change, like let's say we lose Earth's atmosphere, well, fitness goes to zero. And that doesn't seem to quite agree with our intuitive notions of what fitness is, because you want to be able to say you want to describe genomic capabilities in terms of what it's able to do in even hypothetical environments. So the more I'm glad I'm getting this to you early on in your scientific career, because there'll be a lot of things that describe evolutionary literature, and it can be very confusing, and you can end up just going in circles for a long time, just like on the question of speciation. I mean, fitness is right up there in terms of complications, and I'm not saying that as a creationist. I'm saying that as someone who's reviewed what evolutionary biologists themselves have said about fitness. So, so you asked how important is it to our definition? Okay, so genetic entropy 1.0, even with the evolutionary definition of fitness, I think Dr. Sanford was able to argue that there's genetic entropy even for humans. Dr. Dan will make a very good case against it for viruses, and I've said, that's why I said I just avoid all that. I'm not a virologist anyway. I'm a protein guy. But when you add this new variation where fitness is kind of the more common sense thing, in terms of capabilities and hypothetical environments, then it starts to become important. And I don't know if you've heard the book by Michael Behe, Darwin Devolves. Yeah, that uses this kind of modified definition. And the Discovery Institute is pushing this idea of defining capabilities of organisms kind of along engineering lines. And Randy Galuza joins that meeting. I'm part of the engineering group that's sponsored by the Discovery Institute. I see Randy there a lot. So Randy, in addition to being a medical doctor, also had a degree in electrical engineering, and you could see the influence. So in the ID and creationist community, the notion of fitness in the more general sense is being adopted. And I think in the medical world, it's kind of adopted and they don't even realize it. The funny thing, I have to show you this book. Let me see if I could bring it up. And it's called Survival of the Sickest. Let me see if I could show the. It really is a funny title of the book. It's one of the funniest. Let me see if I could get it. Okay, see if it's called Survival of the Sickest. See if I can get a. I'm trying to get an image of it. I'm trying not to get. Okay, I'm going to open. I'm just going to show you the cover of the book because it's so funny. Let me see. Here it is. Survival of the Sickest. This is a New York Times bestseller. And it says, A medical maverick discovers why we need disease. Okay, he's really arguing how natural selection has preserved certain diseases, and this would include sickle cell anemia, Tay-Sachs disease, obesity, cholesterol, and diabetes. And I'm just laughing. And I said, hey, Dr. Sanford, did you know about this book? He said, no. And I said, this is the title of the book, Survival of the Sickest, Why We Need Disease. And he started laughing because the point of this is that the medical community kind of knows that, their definition of fit is like, okay, someone that has sickle cell anemia, obesity, Tay-Sachs disease, and diabetes is not fit. But in the evolutionary sense, it could be. And that's where there's a disconnect. So I think that I'm not really coming up with anything new. I'm just pointing out that I think that evolutionary biology is they're the ones that are actually in left field in terms of what they really mean. The thing that's very, very problematic is, and you could see this, Darwin's Origin of Species, Chapter 6 says, organs of extreme perfection and complication. The evolutionary definition of fitness, and R.H. Brady had pointed this out. He's a philosopher of biology, very great mind. And that's the one we had 10 hours. Emery and I were covering on this channel for 10 hours. Went through that paper. He said, there's no way you can go from like the creature that makes the most offspring makes the most offspring. Okay, that he first pointed out the tautological definition of fitness. The second thing is it's not correlated to the increase of complexity. And what is really bad, as shown by Lenski's experiment, that it's actually anti-correlated. Functional complexity is anti-correlated with selection, and that's really bad. So organs of extreme perfection and complication should not arise. It's kind of only the way Darwin envisioned it is actually only fantasized. It's not reality. And I'm going to show you an experiment. This is really funny. Spiegelman's monster. I appreciate your patience. I'm giving you like 15 years of my research here in just like one sitting. So let me show you Spiegelman's monster. Emery, do you have anything? Oh, maybe he's...
Sorry, I stepped away to get some water. No, I think you're doing a good job, Sal, from what I've heard. I just had to step away for a second. But no, you're good.
Since you're my favorite reader here, I don't know if you could see this. This is Spiegelman's monster.
Okay. Yep.
Can you read it for us? You could save my
Sure.
voice a little bit.
Sure. Spiegelman's monster is the name given to an RNA chain of only 218 nucleotides that is able to be reproduced by the RNA replication enzyme RNA-dependent RNA polymerase, also called RNA replicase. It's named after its creator, Sol Spiegelman of the University of Illinois at Urbana-Champaign, who first described it in 1965. Spiegelman introduced RNA from a simple bacteriophage O beta, or is that Q beta? I can't tell. Into a solution which contains Q beta's RNA replicase, some free nucleotides, and some salts. In this environment, the RNA started to be replicated. After a while, Spiegelman took some RNA and moved it to another tube with a fresh solution. The process was repeated. Shorter RNA chains were able to be replicated faster, so the RNA became shorter and shorter as selection favored speed. After 74 generations, the original strand with 4,500 nucleotide bases ended up as a dwarf genome with only 218 bases. This short RNA sequence replicated very quickly in these unnatural conditions. All right, and then a couple of people have replicated that experiment, except without adding the RNA, only RNA bases and OB replicase. They found that under the right conditions, I think it's QB replicase can spontaneously generate RNA, which evolves into a form similar to Spiegelman's monster. However, Shetverin and colleagues later showed that the spontaneous RNA generation was due to environmental RNA contamination. I can build on Spiegelman's work and produce a similar system further degraded to just 48 or 54 nucleotides, the minimum required for the binding of the replication enzyme. This time, a combination of HIV. Too far, Sal. I didn't finish. Too far. Thank you. HIV1 reverse transcriptase and T7R polymerase. Now you can scroll down.
Okay, what was the point of this? For the longest, the Wikipedia entry got edited because they were saying this is a great example of natural selection. And I think after Behe's book came out, this became an embarrassment. So you could see, look at Origin of Species, Chapter 6. You could look at, again, Spiegelman's monster, and then Lenski's experiment where fitness gains, but the genome decays. And then you remember that one with the E. coli pangenome. Selection tends to go selects against diversity and capability. It would tend to resist evolution of organs of extreme perfection and complication. I mean, I know the evolutionary biologists will debate me on that, and I'd say, you're getting the weight of evidence against you at this point. I mean, it's getting harder and harder. That's why I'm still a creationist. I'm just like, I've seen this trend, and I see you're smiling, so I hope you like what you're hearing.
Yeah, no, that explained it. I think that's a very interesting example.
Oh, thank you. So these are kind of like the general principles. We are kind of focusing on human evolution, but I gave you kind of a lot of other general principles to think about. I wouldn't necessarily call it genetic entropy. I would just, what's in a name? We have all these concepts, but I think one, mutation will degrade the genome, but then natural selection also can contribute to degrading, because it says genome decays despite fitness gains. Fitness gains is natural selection acting, at least Darwinian selection. I won't call it natural. That's a misnomer. Darwinian selection. Because if you call it, so I like Darwinian selection. So if you don't have any questions, I'm going to just go on with a little bit the rest of the presentation. And, once we hit about 2 hours, we can call it a night, and you can munch on this and we could have a further conversation. And then you can also talk to Dr. Dan and have conversations with him. So did you have any comments or questions before I go on?
No, I didn't. Go on.
All right. So, I really appreciate your audience, because this gives me a chance to articulate 15 years of work.
It gives you a chance to be a nerd, Sal.
Yeah. Okay, so why was the junk DNA hypothesis? My answer is the idea of large amounts of functional DNA is incompatible with evolutionary theory. So remember, we can only tolerate if every human child has at least one mutation that the parents didn't have, one additional. The genomic decay is irreversible. And there's some mathematics associated with it. It involves the Poisson distribution. That's like our lecture. But let's see. Let's see what Dan Grauer had to say. He said, studies have shown that the genome of each human newborn carries 56 to 103 point mutations that are not found in either of the two parental genomes. If 80% of the genome is functional, as trumpeted by ENCODE Project Consortium, then 45 to 82 deleterious mutations arise per generation. For the human population to maintain its current population size under these conditions, each of us should have, on average, 3 times 10 to the 19th or 5 times 10 to the 35th kids and children. This is clearly bonkers. If the human
that's a big
genome,
homeschool family.
Huh?
I said that's a big homeschool family.
That's a big homeschool family.
I'm sorry.
Thank you. If the human genome consists mostly of junk and indifferent DNA, if the vast majority of point mutations are neutral, this absurd situation would not arise. Now, what's really funny about Dr. Grauer is, as he started to see the evidence mount against him, he's changed the figure a little bit of what could be tolerated. But again, he's using the evolutionary definition of fitness. If you use the one that I had suggested that's based on capability, this figure would still hold. So I'm just totally ignoring his latest papers. I said the guy's just changing his mind, and no one wants to be his co-author anymore on some of his diatribes promoting junk DNA.
So basically, this would be arguing for a human population to maintain this population size for the foreseeable future.
Okay, let me explain that. Do you remember that picture of the Punnett Square where you had to have like, what was it, 16 different genotypes, and then only one is, that red, that green wrinkled one? You can apply the same body of math, and you're going to need these 10 to the 35th power genotypes, and only one of them will not have the harmful mutation. It'll be clean. So every female needs to make 10 to the 35th kids, and one of her kids will be clean. You have to assume then the natural selection to come along and kill all the, 10 to the 35th minus 1 kids. So you need 100% efficiency in selection, and you need the mother to have 5 times 10 to the 35th kids. Poor mom. And there was a funny moment when Dr. Sanford gave his lecture. This is so funny here. Let me see if I can show it here. He said, okay, so he first quoted, I think, Dan Grauer here. In the average human genotype, over 100 genes are dysfunctional or altogether missing, and over 1,000 genes are substantially impaired. That was by Alexei Kondrashov here. He was a researcher at the NIH. His colleague was my professor at the NIH, FAS graduate school at the NIH. And there's another quote here. A newborn human carries about 100 new de novo mutations originated in the germline of their parents. About 10% are substantially deleterious. And okay, let's say that we had, see if I have this. Okay, let's say we only had 1% of the gene, okay, let's say we just use the figure 10% of the genome's functional. Well, which Dan Grauer would say that about 10%, he'd like to have it around 10%. Well, even if we use Dan Grauer's figure for how much is functional, let's say 10%. Let's say 90% is junk, 10% is functional. Well, we pump that through the equation. Again, this is based on the Poisson distribution. It's an 1.5-hour lecture on how we calculate this. We would need 99.95% elimination. So the mothers would only need to make 44,000 offspring instead of 10 to the 35th. So if ENCODE is right, so the point is, Dan Grauer is criticizing ENCODE, and he was saying you need 10 to the 35th. But if we use Dan Grauer's own numbers, we only need about 44,000. But still, these are outrageous numbers. And that was a really humorous point in that lecture. It's like, Dan, even if we accept your numbers, you have no reason to be railing against ENCODE because each female is still going to be needing to make all these offspring, like 10 to the 11, 44,000 offspring. And if we have like, I think that's with like, 10%. If we use Dan Grauer's figure, it's like 10 to the 11th, which is a huge number. And ENCODEs would take this up to 10 to the 35th. And so that's why I was focusing so much on the non-coding regions and about that RNA phase separation. We're trying to explain what the role of these transcribed RNAs are. And it would lean toward justifying this ENCODE figure. But even just say 10%, that's an accepted figure. Even the evolutionary biologists will give you 10%. You're going to have genetic deterioration if you use the conventional notion of fitness. It's going to be inescapable. And if you ever want to go through the math, I can walk you through it. It's tedious. It's just a little painful, but you could see it. And so Dan Grauer said, if evolution is right, if ENCODE is right, then evolution is wrong. And I'd say evolution's already wrong, even if we use the most, most generous figure that 90% is junk. It still would fail. You can go up to 99% and it's still problematic. 99% junk, and you still have this problem. And so Dan Grawer was pretty mad at the ENCODE consortium. He called them ignoramuses, crooks. He called the leader the scientific equivalent of Saddam Hussein. He called the director of the NIH, Francis Collins, a creationist. He was livid. Emery.
Yeah, just sum up how Grawer feels about ENCODE. He literally wrote a paper that was partially titled "The Evolution-Free Gospel of ENCODE." Yeah. He's not an unbiased source, but even his numbers have significant problems.
Yeah, traditional evolutionary bio people are not that interested in this line of thinking. I think that is fair. They're more interested in phylogenetic trees. They love, oh my goodness, they love phylogenetic trees. They get along well with the Biermannologists and building all their little hierarchies. That's not exactly right, but okay, they're not interested in this line of thinking. The population geneticists who are part of the evolutionary community, who are really deep into the math, that's where this interest, and there's always been a disconnect between kind of what I'd say the mathematical biologists, population geneticists, versus kind of your typical evolutionary biologist, because a lot of the population genetics types are mathematicians. They're not your traditional biologists.
So basically, how do they try to get around this, right? They either are going to have to somehow require some sort of selection to take those out, or they're going to have to deny that figure. What's kind of the general kind of response to that?
There's a variety of responses. So for human evolution, they all agree we're deteriorating. They don't like to kind of put two and two together and say, well, then we couldn't have evolved in the first place. They leave that to someone else to figure out. So the general response is like, well, that's not my area of expertise. Okay. That's I think that's an honest enough answer. It's like, it's their way of saying, I don't know. I hope someone else will figure it out. And that's been, that's kind of the way I perceived it. It's someone else's problem. It's someone else's problem. Evolution's fact. Someone else will figure it out. Okay. So that's been their response. But where we got some of this stuff for Dr. Sanford actually came one of his genetics professors. He attended the same university where James Crow taught, but I asked him, I said, did you ever actually study under James Crow? He said, no. He said it was a different department. But this idea has been floating around, and it's always someone else's problem. Their first line of defense was always to say that as far as humans are concerned, the genome's mostly junk. And that's why you keep hearing that word junk DNA. And they're kind of trying to I think the evolutionary community kind of wants to make that term be forgotten because it's an embarrassment to them. And I'm trying to keep it alive to just kind of remind them where they screwed up.
So basically the idea there being that if so much of the genome is junk, we can have all sorts of mutations happen that don't actually degrade the organism genetically because the DNA is non-functional.
Yes, that was basically neutral theory.
Right. Well, let me say this. It would have to be 99% junk. And then they have to use their evolutionary definition of fitness, because then they can add these things like renormalization that Walker and Keatley threw in. I mean, there are all these equivocations of what fitness is, and they can kind of like redefine it on the fly. And that's how they got around it. That's so in answer to your question, how did they dealt with it? So Dan Grawer's latest paper and then Ira Walker Keatley kind of came up with a clever mathematical way that if you use the evolutionary definition of fitness, you can at least avoid extinction. And so, but they covered up the problem of genomic decay. So remember, the genome can decay despite sustained fitness gains. So the first thing they did is they just used only the evolutionary definition of fitness. They didn't have to deal with the loss of complexity or information. That was the first line of defense. The second line of defense was with the evolutionary definition of fitness, they just kind of they figured a way that they could reduce the impact of a deleterious mutation by just kind of reducing its effect on the reproductive rate. And then you can get around it. And it's like, okay, you can imagine that, and maybe that will work. At least it doesn't go extinct. But you've done that by covering up the real problem of genomic decay. You've given up Darwin's vision of organs of extreme complication and perfection because you've just totally decoupled the definition of fitness from complexity. So that's the next line of defense. Because the original line of defense was the genome's junk, and that's becoming untenable, and it's very unpopular. So when you go to evolutionary literature that tries to address this, you'll just see pages and pages of math, and it looks impressive. It's all theatrical. And I can walk you through some of that, but what's really funny is because I studied math, and so did Bill Basener, who happens to be a professor emeritus. He's already retired in mathematics. And Ola Hossjer, who's an award-winning mathematician, we all knew that this was mathematical theatrics. I said, you can't pull the wool over our eyes. We know what you're doing. And so we know that their response is the best that they could make given the circumstances, but it's fundamentally wrong. And that was kind of the basis of a little bit of the paper that got published in the Springer Nature Reference work.
So what I kind of hear referred to often next is the idea that, right, we've been cultivating lots of different organisms for many, many generations. And why aren't we seeing this, depletion, basically, of genetic information in them? Now, I know, I think Sanford has argued, based on some papers, that we have witnessed that. Would you argue that for other organisms other than humans? Well,
okay, I'm just going to put out there, if you don't think a poodle is degenerate, I don't know what I can do to help you. They're cute dogs, but they wouldn't survive 10 seconds in the wild, and they come loaded with diseases and goodness. I mean, a lot of domesticated breeds are like that. They'll do fine in captivity where they get constant care and everybody takes care of them and treats them real nice and whatnot. But they don't do so well in a wild condition, which creates, because they're not subject to the environment, they're not subject to selection really, except for things that people want. So it's kind of comparing apples and oranges, because you would expect something that is receiving the best possible human care and attention to live longer than and survive better, even with lower genetic diversity, than you would something living out in the wild that has to find its own food, find its own home, find its own mate. All those things take energy, take risk, has to avoid predators. Your little Chihuahua doesn't have to do any of those things. And it's, it gets to sleep in the air conditioning all the time and, yip at people and bite people in the ankles. I mean, it doesn't really actually, and no, I don't dislike Chihuahuas, by the way. But it doesn't really, again, you're comparing apples and oranges. You're looking at organisms that are basically sheltered from womb to tomb, compared to organisms that actually have to fight for their own survival. So there is a significant difference. And Sal, if you want to add to that, go ahead.
No, I don't have anything to add. I forgot. I'm having a Joe Biden moment here. But what was the question?
So I was saying, right, we've observed a lot of different populations of organisms, like cultured bacteria and certain types of mammals as well, right? And you talked a little bit before about why you think, it might not work the way it does in humans for, viruses and possibly for bacteria because of certain interactions they have. Is there a reason why we wouldn't detect it in other mammals, like perhaps mice? Or have we?
Okay, wait. See, this is actually a really good conversation, and I told Dr. Sanford one of our first meetings. I said I'm very concerned about the problem of mice, And so genetic entropy 1.0 was very insistent about the human genome decaying and we would die. That still may be true, but then we have the problem of mice, and they're obviously alive and they've had a very high reproduction rate. And that's kind of been a thorn in the side of the genetic entropy hypothesis. So if we use the original definition of fitness that's tied to reproductive rates, and then we say most mutations are deleterious, yeah, you would eventually get the mice dying.
Mm-hmm.
But what I think is happening is they've just lost, they continue to lose versatility, but they're able to still thrive. So we have examples of like organisms that are just losing so many organs and capabilities and they're still alive. So at the bacterial level, definitely, I showed you the pangenome. You could see it's lost 80% of its genome and it could still possibly be alive. It doesn't go extinct. It doesn't necessarily go extinct, but it has genetic decay. So what I call for genetic entropy 2.0, I would not tie it to extinction. It's very hard to tell when a species will go extinct. I mean, it's...
So that's a fairly big difference between you and Dr. Sanford then?
Yes.
Okay.
Yes. And if we co-author a book, I don't know. I'm just going to assiduously avoid that topic. Because try to time how long a species will live. Okay, hypothetically, if you did this mathematically, and you could estimate the average effect of a deleterious mutation, you could predict when the species is going to go extinct. And I think that's a really tough road to go down. One reason I'll point that out is there have been experiments with yeast. When they had separate lineages, 2 of them would die. Why did the others survive? Maybe just a little turn in mutations, like say the genetic repair mechanisms, could affect the entire trajectory of how it's going to thrive or not thrive. I don't know this for sure, but some lineages of Lenski, I think, are at risk of actually going extinct because they've lost their genetic repair mechanisms, and they may actually melt down. So the problem of extinction, I'd rather not go down. And I felt Dr. Dan made a good point about that we ought to, this should predict extinction. Well, that's why I opened the show with that quote from Bertrand Russell: we will all go extinct. So it's guaranteed. It's just how and when. And I'm leaving that as an open question. So that is a difference. You are correct.
What do you think of kind of Dr. Duff's point here? Because specifically for humans, right, we have a pretty extensive genetic record, and obviously we can tell mutations that have arisen. Do you know of any possible correlation between the number of mutations that we've gained since kind of these ancient, some of them ancestral human populations, and if it would be possible to match that to, for example, like the exponential curves that genetic entropy would predict?
What was his specific question? He has a lot of comments in the.
Yeah, so he said something about horses, and he said, I don't see how there is any genetic entropy evident between their genomes, that of the modern horse and that of a very early horse.
I don't know about that. Can I just throw this out? Genetic entropy for humans is testable. We may not be alive to see it, but theoretically, hypothetically, 100 generations from now, it'll be pretty clear who's closer to the truth. So maybe the Lord will return by then and this will all be moot. But I do want to point out this is testable. And we can also, for one, our sequencing methods are a little bit, they're limited. Are you familiar with how sequencers work? Some of the cheaper ones? Just the chemistry of it is that the genome gets chopped up into 100 nucleotide strips, and then we have to reassemble it. And then we do that by throwing it on a template, which may cause some problems in the accuracy. And Rob Carter said, I would rather we have less sequences, but more quality, because we don't have quality. I think Nathaniel Jeanson and Dr. Sanford would be there. But the reason I throw that out is we do have generations. We have grandmothers and we have great-grandchildren. Even within those 5, I think we can detect genetic deterioration. So, I mean, how far do we have to carry out this experiment to start to make conclusions? We, you know, in theory, we could test this and we could show it. And I've already shown, I've just quoted evolutionary biologists like Dan Grauer, who hates creationists. He's an ardent atheist. He said his estimate is 100 per generation, 100 mutations per generation. So if most are deleterious, either functionally or whatever, and that's why I went into the RNA phase separations, you take his stuff and you just keep adding it, and it's like there's been genetic entropy. I wouldn't, you know, I mean, the human genome is what, 3.3 gigabases? So you multiply that by what, 72 generations? It's still maybe only 7,000. It's still a small fraction if we're talking like, say, point mutations. But I think the translocations could be very serious, meaning you have huge segments of the chromosome being deleted or repositioned. That could be really, really serious. That can disable a lot of genes. I will tell you this, in domesticated grapes, my professor at FAES NIH, she gave me this thing about 15% of the genomes of grapes, they've already lost one of their, one parent has already lost an entire gene. Emery would know the correct term for that.
So basically, your argument here would be the reason why we aren't detecting this genetic entropy between us and some ancient humans that we've observed is because the DNA is unreliable, you think, or at least our sequencing of it is.
I would say that. I would absolutely say that. And the other thing is we don't have, you know, there's still a lot about cellular biology we don't know. That stuff on the RNA phase separation has only come out in the last 4 years. We would have, you know, a lot of people are like, well, you have two camps. One will say, we don't know what it does, therefore we'll just assume it's junk. And the other camp will say, we don't know what it does, but we'll assume it's there for a reason and it's functional. And it's experiments that'll decide. And so how we could say that there's no genetic deterioration, that we don't have any benchmark for saying that. We can say our best science would predict that there should be we should have accumulated, you multiply 100 roughly by the number of generations between us, we should have accumulated at least that much in point mutations, but we don't know the number of translocations. You know, that means huge, like 10,000 genes just put somewhere else. I'm sorry.
Sorry. So you would say that hypothetically, if we could get, you know, the correct genome, that we would be able to see these effects, you know, from some of these early humans that we've sequenced it. We should be able to detect that.
Oh, yeah. Now I know what I
decay.
wanted to say. Remember I said about the sequencing technology going only 100 bases at a time? That makes it very difficult to detect translocations. So let's say you get one 10,000 chunk of DNA and it's put in a different location on the chromosome. So they call this synteny differences. Now that we know about the 4D nucleome, it's really important. So our sequencing technology is not quite good enough to detect those large scales. So I don't know how we can get a Neanderthal genome and see the synteny layout. And let me just show, to me this was, okay, let me show you something. Synteny.
While you're looking that up, so do you think that if, for example, these ancient human genomes that we have, if those are correct, do you think that they would disprove your model? Or at least
not
disprove
necessarily.
that
I'll tell you what would disprove the model.
All right.
Okay, the falsifiable hypothesis is the genomes in the future. Okay, so what I would try to say is let's not use, we can use genetic entropy. I I wouldn't even call it genetic entropy. Let's just say some of the pieces that we've studied tonight. The natural selection selects against diversification of, it selects against versatility, and hence organs of extreme perfection and complication shouldn't emerge. So one thing about the genetic entropy hypothesis, it's trying to say, we should all be dead, therefore we didn't evolve. And I say, let's just try to make something not quite so ambitious. Let's just say selection it selects against emergence of complexity. I think that's more defensible than trying to go to extinction. So I didn't get your question answered direct on, because I think it is too hard to, this is where Dr. Sanford and I might disagree, is he will insist on headed toward extinction and I wouldn't.
Mm-hmm.
But I would say, I would still say that he's come up with enough both intuitive and mathematical theory that we could say things just don't evolve naturally to organs of
Sure.
extreme perfection and complication.
What
And
I'm
that
kind of
there's-
struggling to understand here is exactly why, if we could, have a high confidence sequence that we created that we knew was likely to be correct, why not observing genetic entropy wouldn't disprove that model?
No, I don't think I'm answering your question. Let me just think about that. All right. I would
Right,
first
because
say,
from what I'm thinking, right, we should be able to match that right onto the exponential curve, right? If your model is correct, right, those sequences, we should be able to match to the curve. Am I thinking wrong, or is that?
the one thing is though, you can't see that exponential. Okay, so the point mutations independent of translocations. Okay, let me first show you this picture here. I really want to show you this picture. And this is important when we're talking about sequence technology. So it says here in genetics, the term synteny refers to two concepts. In classical genetics, synteny describes the physical co-location of genetic loci on the same chromosome within an individual or species. In current biology, synteny more commonly refers to collinearity or conservation of blocks of order within two sets of chromosomes that are being compared with each other. I'm sorry, that's really confusing. Let's go to a picture. So if we take the mouse, I think you could see my cursor here. See on the right here, this is the mouse.
Yep.
And you can see the color coding.
Yes.
You compare that to the humans. So if you're mapping, you can see where the light green here is on like chromosome 10. Look where all the light greens are. Okay, and you could just do that for all of them. So why did I bring up the problem with sequencing? What if we have all these between individuals? With our current sequencing technology, it'll look like we're all about the same. But what if the humans are radically different in terms of their layout on their chromosomes? That could make a difference because we're finding out the chromosome locations actually could be very important. That's because of what we call the 4D nucleome, where gene regulation is dependent on physical geometry on the chromosome. The layout is actually important. So
now what's typically going, oh, sorry, go ahead.
So I'm just saying, I think that hypothetical one would be difficult first. And I want to just say that that would be a very large hurdle to overcome.
Okay.
And given we have so little of the, Neanderthal samples, it'd be difficult. But secondly, going back to that curve, that exponential.
Well, especially since Neanderthals aren't necessarily, they're not ancestral to Homo sapiens, right? So we would need to use Homo sapiens samples, not Neanderthal samples.
Okay, Homo sapiens samples. Okay, I stand corrected. Genetic entropy ages.
I mean, like, they did interbreed with us, so I guess, like, you might be able to detect it, but we only have 20% in all of humans. So, yeah, it would
Okay,
probably
so
need to
let's
be
look,
sapiens.
okay, so let's go back to this exponential curve you were talking about here. Would we see an exponential difference between, say, a patriarch and today? I would say no. It would be a linear difference in genetics, but the effect, I don't know how we can calculate that the effect would be exponential. So let me just try to explain this. Let me show you, this is actually, let's see. I'm going to show you something very important. Early aging disease. And this was in my progeria. Okay, I'm going to tell you one thing. And respect to these people, I'm sorry I have to bring this up. This is scientific, but I just feel for these kids, all right? So progeria is early aging. These people live, let's see, progeria is a specific type. Okay, let me see. Okay, so the prognosis, few people with progeria exceed 13 years of age. 13 years of age. And I think it's actin. Is it actin? Lamin A. Okay. All right. So I'm trying to answer your question, and I'm sorry I have to go through so much to get there. We have 20,000 genes. One gene that is disabled can cause an age change by a factor of 80% loss of their age. One gene out of 20,000, and that gene is lamin A. A-type lamin gene. And actually, let me see the size of this. Uniprot.org. I'm just going to look for lamin A, the size. Lamin A. Lamin A in humans is only 664 nucleotides. I mean, amino acids. So that would translate into what, 1,900 nucleotides out of 3.3 gigabases. You disable a few of it, you can actually kill the gene with a premature stop codon. You might have as little as just a few nucleotides, you could kill the gene, just disable it. So even that small change then can create progeria. So let's just extrapolate this to the loss of age. And just using rough figures, it wouldn't take but to disable 2 genes, maybe even just a few nucleotides, and we could go from living 1,000 years down to 100 that easily if the right things are knocked out. So when God pronounced that humans will not live to be 120 years, more than 120 years old, okay, there's some interpretations of that don't talk about the length of life, but let's say that that's the interpretation. Mm-hmm. And I think I could make a good case that is the correct, that God is putting kind of a expiration date on every one of 120 years, minus some exceptions like Moses. You wouldn't need to knock out a lot of nucleotides. So you can't really correlate it to the number of nucleotides knocked out. It's not the quantity, it's the quality, so to speak. Which ones were actually knocked out that could change on a dime, going from long ages to short. So the idea of reconstructing the human genome, we would actually have to know specifically which locations changed. It could be as few as even 1 or 2 nucleotides if it introduces a premature stop codon. So We really don't know.
Okay. So basically, though, based off of like the number of mutations, could we possibly hypothetically form basically the number of mutations which we have relative to this hypothetical individual and try to basically construct that or not?
If we could get the synteny in line, then I think we would have a better chance. But I think people are really underestimating the problem of synteny.
Okay. Now, the general kind of response to the view that these are sampling errors is typically, right, that we can line them up, that they match, right, that we can form clades out of them. Do you have kind of a response to that?
Can you repeat the question?
Yeah, so generally, like when we're talking about the Neanderthals or like a specific group of Homo sapiens is very ancient, when people say they're, basically we're sampling them wrong, or there's, some type of problem with the DNA, we usually turn to the fact, right, that we can form clades out of them, or, that we can find that different individuals are very similar to one another. So typically that's understood, right, to mean that this is not a sampling error. But is there a way in which like a sampling error like you're discussing could cause us to still end up with a neat clade, or with these individuals still looking very similar, even though we've sequenced them both wrong?
I think you're far more expert at that question than I would be able to answer. I actually think you would be far more knowledgeable at this point. I don't have a good answer.
Okay.
Can you explain? If I'm understanding this right, it seems you're really interested in reconstruction of the past, and you want to compare. Okay, all right. Okay, I told you I'm not into paleontology because of the frustration of trying to do reconstructions. And can we just pause a little bit, just take a little bit of a diversion?
Yeah, absolutely.
You had said that you're into model building and I'm into model burning, so to speak. Meaning,
well,
I like burning down evolutionary models.
Pretty important.
I'm sorry?
I think they're both very important. Really, they kind of go hand in hand.
Right. So you said you're interested in what's true, and I'm interested in destroying what I think is false. And the reason I like doing the way I do it is because it's a lot easier. Trying to reconstruct the past is, I'm interested in the truth, but it's easier to show what can't be than what was. Mm-hmm. So your job, I think, is a lot harder, and I kind of like the easier route. And it's more conservative because if you're trying to build a model, it can be easily falsified with new data.
Yeah.
Whereas if you're just criticizing a model that's wrong, you just pile on like abiogenesis or eukaryotic evolution. It's like you're not going to be overturned. Go on, Emery.
Well, I was just going to point out that that as someone in the scientific field, and I can't cite this appropriately, so forgive me if anyone in the chat knows the correct citation. Feel free to drop it in. But all models are wrong, but some are useful. And that I find the more I dig that this is true. Like quite a few models that get thrown out there are wrong, and everybody knows they're wrong, or at least everybody in the literature knows they're wrong. They just go, well, they sort of help us right now. Maybe we'll move them later, but right now we don't have anything better. So, yeah. I'm sort of I kind of walk a middle line here between Sal and maybe where Peter is. I like burning models down because that's fun. I never quite got out of the teenage pyromania phase. So I like watching the bad ideas burn to the ground. Yes, thank you. That's the quote I was referencing. But I also want to put something up in its place that is going to actually function. And again, it may not be perfect because all models are wrong, but some are useful. But at least it's going to be better than what was there previously. Thank you, Dr. Duff. So again, So I kind of walk a middle here, and I don't do too well with reconstructing the past. I don't know so much about that. I stay out of human evolution almost entirely, just because I don't like people. I'm legitimately serious. But I prefer animals and plants and that sort of thing. So I get more interested in that. But I think there's a place for both. I don't really like the way that
sometimes it's presented as a dichotomy, but it's
one
really
likes to build a model and one doesn't. Go ahead.
Oh, I was just saying sometimes it's presented as a dichotomy, and really it's not. I mean, Sal, for example, here, I mean, building, kind of trying to expand on this idea of genetic entropy. I mean, he is building his own model. I mean, however much he says about not liking model building, that is what he's doing.
Yeah, and to a certain extent, even model burning is building a model, because you are building a null model, not this.
Yeah. So my talk at the Creation Research Society will be somewhat of a model burning talk. I don't know what the board will think about that, so maybe that might be my last talk.
I don't know. I don't know who all's on the board, but I mean, I think that at least some people on the editorial board would be open to model burning. I don't think Clary or Leitner will have a problem with you burning down a bad evolutionary model.
Or a bad creationist model, like there's no geological column, or I'm sorry.
Yeah, I'm going to be doing a paper hopefully at the Origins conference, and I'm going to be doing something connected to human origins and burning some creationist models. That'll be very controversial.
Well, if it's I think if it's done in a spirit of academics, it can fly. And I know that you've had some different views than Dr. Sanford and Chris, and soon to be Dr. Roop.
Yeah.
And that's
Ooh. Didn't know he was getting his PhD. Good on him.
Yeah, good on him.
Mm-hmm.
Hey, and while you're here, Dr. Duff is here. I already related the story. I did go to bat for you, Emery. I was thinking, that'd be glorious. You're Dr. Duff's student.
That would be funny. That would be legitimately funny. I'd get a good kick out of that. I don't think he would, but I would.
That's probably why burning down evolutionary models at least can endear you to most of the creationist community, because you don't attack someone else's. But I recently had to just disagree about Jason Lyle, and that was kind of painful. I mean, I'd be forgiven for that for a long time, but I'm just like, well, that is, after all, going to be kind of one of my. I'm kind of evolving out of biology to go back into physics, so I'm leaving it to Emery and guys like Peter. I'm sorry.
I said, for shame, you're a good biologist.
Oh, God bless you. the
this
bad
is
thing
more
is,
along the lines of, I don't have anybody to ask about my evolutionary biology homework anymore. I'm not even joking. I took an evolutionary genetics class as part of my graduate degree, and how long did we spend on that homework? You trying to explain what they were talking about?
Well, I just happened to be lucky that I happened to see that formula elsewhere in one of my published papers, and I was like, oh, that's what it means. And it's like, I would wonder if anyone else in your class got it right except you.
The class was not very large, and the professor told us what the average grade was. It was not very high. It was pretty ugly. It was difficult, and even mine got docked a little bit because I didn't do the formula notation quite right, and I'm like, okay, whatever, I don't care. I spent 5 hours on this homework assignment. I don't care. I just want it done.
And yeah, so one of the people in the chat, Arn Craigbaum, he asked, does the evolutionary community go to this line of thinking? And I said no, because most of them aren't mathematically sophisticated. And that's an example right there. Taking a genetics class, and most of the class had a low grade because the math is not, it's not easy. And to make matters worse, the evolutionary literature doesn't really help you understand it. You have to hack at it for a while.
Yeah.
And you realize, oh, that's what it means.
Yeah, because I mean, it's specialists writing to other specialists, which I don't blame them. That they don't expect that graduate students are going to be sitting here trying to fumble through their papers trying to understand what they're talking about. So they don't dumb things down enough for the rest of us to understand. So I'm not necessarily blaming them for that, but I will grant you, I've tried reading some population genetics papers and gone, oh my head hurts.
I know we'll get back on topic a little bit, but I just got to give you an anecdote, and I told you this before. Maybe Peter will laugh. I was driving on my way to church, and I was so fascinated by Emery's textbook when he's going through the homework. I said, "That's a really cool textbook," and I'm like, "I never saw that in what I was reading." I said, "What does this mean? What does this mean?" Oh, it's heterozygous advantage, sickle cell anemia. That's what that formula describes. I was so excited. I wanted to go back and give you a phone call. I said, Emery, I figured out what that formula is actually describing. It's sickle cell anemia.
Yeah, it took us a while. We had several hacks at that because it was just very. They worded it kind of funny, which didn't help. But the math is very difficult. If you want to go to PopGen, the math is very, very difficult. Which is why I don't go to PopGen.
And you know what's really sad? Most of it's garbage anyway. I'm sorry to say.
You're allowed to say that. You're the mathematician.
Well, okay. Maybe I could discourage Peter from doing. Let me just give you a little discouragement not to go into population genetics because
I don't plan on it.
Okay. Well,
I do not like math very much.
Man after my own heart.
All right. So let me just share this briefly. See if I could find it. They had this big thing in population genetics on the Ka-Ks ratio. In genetics, the Ka-Ks ratio, also known as omega or DN-DS ratio, is used to estimate the balance between neutral mutations, purifying selection, and beneficial mutations acting on a set of homologous protein coding genes. It is calculated as the ratio of the number of non-synonymous substitution per non-synonymous site, Ka, in a given period of time to the number of synonymous substitutions per synonymous site, Ks, in the same period. The latter are assumed to be neutral. So Ks is assumed to be neutral. So all this evolutionary theory is built on Ks being neutral. And I shared that in the last month, there's a paper in Nature that said it's not neutral. We found it in yeast. That the overwhelming majority are deleterious. I said, oh my goodness.
So much
maybe
for
hundreds of thousands of paper has been falsified.
Yeah. That was what hit me too when I read that paper. Is like, well, if this is correct, goodbye neutral theory. and, but yeah, no, it again. I'm hesitantly. I don't want to jump the gun too much on it because it looks good, but one paper does not make a scientific body of literature. I'm looking to see like another 5 come out before I get too excited about it. I think it's interesting, but I don't want to start drooling yet. I'll start drooling when you get about 5 or 10 papers out there saying roughly the same things, and then at that point,
true. So let's just make it a gentleman's bet here. But if that paper turns out to be the first among many, this is what Huxley said. He was Darwin's bulldog. He said, the great tragedy of science, the slaying of a beautiful hypothesis by an ugly fact. Let's say a single ugly fact. And that could have been the ugly fact that slayed hundreds of thousands of evolutionary biology papers. Yeah. Okay, so we've had our fun. If you want to wrap up, oh my goodness, we're almost 3 hours here. So I promised we'd end at 2 hours, and we didn't. We had a great time. Do you have any closing questions here, Peter? And you could save them up. We can have another session sometime. I've had a great time, by the way.
Yeah, it was great. I think I learned a thing or two. Very informative. I kind of asked you the questions I had for now, so I don't really have any at the moment.
Okay, let me just go
Sorry,
back to
but-
my slideshow, see if I caught anything of interest, and we'll wrap up. So okay, so that's Dan Grauer. You'll hear that term. And he was very much against the ENCODE project. And so the papers you've been hearing me read have kind of really been embarrassing to him because we're finding a lot of function that he said doesn't exist. So the cellular biologists are finding this. And oh, good night, girl. Good night. Thank you. You're an inspiration to me. Good night, girl was accepting orthodox evolutionary theory, but as nice girl studied this more, became convinced of ID on some level, and I'm extremely grateful for that. So let me just see what else I had. So even okay, so one thing I'll point out, even in the same university, there's been a conflict between professors working at the same university over junk DNA. Stamatoyanopoulos versus Felsenstein. Here are some other headlines: contamination in the genome by very slightly deleterious mutations. Why have we not died 100 times over? So that was a colleague of my professor at the FES NIH. And he really didn't answer the question. He came up with some mathematical thing that I said is kind of a trick, and I don't think it's correct, even shown experimentally. So he wrote the book Crumbling Genome. Adam's Curse, I showed you that one that had that exponential decay curve, so that's another name. Lawrence Lowe, I find that a surprisingly large range of biologically realistic parameter combinations would lead to the extinction of the human line over a period of 20 million years if accepted wisdom about mitochondrial DNA and Mueller's ratchet is correct. And that's just empty DNA. So here's an example of, okay, so this isn't a yet timeline. I think if you relax that requirement, then I would kind of lean toward the extinction thing of John Sanford. And let me just say this, just kind of comically. I was saying, hey, John, the environment can change fitness, so genetic entropy can be due to cataclysms. I said, why can't we include that in genetic entropy? Because then we could even include, Bertrand Russell saying the universe is going extinct, therefore genetic entropy is true. And Dr. Sanford said, no, I kind of like the self-destruct model better. So that's kind of a paraphrase there. I just thought that was funny. He likes the self-destruct version. So let's see here. So I'm glad I'm sharing this with you, just so you know that this is mainstream. So if you get, the evolutionists will challenge the viral model, and I'm just going to grant that, because the real issue is humans as far as you and I are concerned. If we are like Drosophila, the decrease in viability from mutation accumulation is 1 or 2% per generation. This is more than compensated by much more rapid environmental improvements, which are keeping well ahead of any decreased efficiency of selection. How long can we keep this up? Perhaps for a long time, but only if there remains a social order that permits steady environmental improvements. He's saying our medical technology has to keep improving to compensate. And he's one of the most famous, while he was alive, I'd say he was the #1 population geneticist. Here's one: risk of population extinction from fixation in new deleterious mutations. And that was in 1994. So, and here's Crabtree. I would wager that if an average citizen from Athens of 1000 BC were to appear suddenly among us, he or she would be among the brightest and most intellectually alive of our colleagues and companions, with a good memory, a broad range of ideas, and a clear-sighted view of important issues. Furthermore, I would guess that he or she would be among the most emotionally stable of our friends and colleagues. And again, I pointed out the problem of trying to reconstruct from ancient genomes, we would have to target specific. Like I said, lamin A is enough to cause 80% loss of age. Just that single 664 amino acids. So anyway, but it would be nice if we could figure out how to make ourselves better genomically. So this is pictorially. Look at all the scientists here that say that the human genome is deteriorating. And when I was in the debate with Dr. Dan, I specifically asked, do you think the human genome is improving? And I couldn't discern what he had to say. And that if we talked again, I would just pose that question. Do you think the human genome is improving, not in terms of evolutionary fitness, but the idea of decay, losing genes, losing versatility and capability? And I said, look at all these very respected geneticists. They're all secular, and John Sanford is a great secular geneticist. There he is with his secular invention, but he's a creationist and a secular geneticist. We have a large number of people. I mean, James Crow is like one of the most he was the most respected in his day. Michael Lynch is very high. And Herman Muller, who brought this whole thing about genetic deterioration, he was a Nobel Prize winner. So this is kind of in the broad sense of human genetic entropy. This is I think it's very well established. You'll be on good ground. If you get into the stuff about bacteria, mice, and viruses, I think that's where even I would have, I wouldn't feel comfortable defending that. So let me just close. If you had anything else, I'm going to close with something here. Peter, Emery?
Yeah, it was great to hear a little bit about genetic entropy from you. And great talking with you too, Emery. So
yeah, glad to officially meet you. I think I'd seen you around, but I don't think we'd interacted very much at all. So.
Yeah, we'll have to talk again sometime.
Yeah, definitely.
Yeah.
I mean, I'm buried under graduate school, but I try to make time to show up when Sal wants me to show up. So
yeah, Peter and I have kind of bonded because we're kind of being persecuted by fellow young Earth creationists. But don't worry. Yeah. Hey, I just wanted to
know about that.
I just will say this: if you become a model burner, you'll be able to get along with a lot more creationists. When you start attacking other creationist models, it's like, well, yeah, that's kind of rough. I mean, you are getting toward the truth. Just a little thing about the politics of it all, because
yeah,
there's so many young Earth creationist models for distant starlight. I guess we're all going to be just feasting on each other, criticizing each other.
I stay well out of that. I don't know near enough physics to even dream of commentating on that.
So let me close for you, Peter, and everyone in the chat. I want to give honor and glory to our Lord Jesus Christ. This was at the end of Dr. Sanford's book. So I feel that we've established well that the human race is going extinct. It's just a matter of when and how. So this is Dr. Sanford. It's very touching. He said, "When I was young, I accepted the fact that I was going to die, and that all of the people I loved were going to die. I accepted it, but it robbed me of joy, to say the least. I was taught that there was still one hope, that the world was getting better. Science was advancing, culture was advancing, even mankind was getting better. Through our efforts, we could make the world a better place. Through evolution, we could evolve into, through evolution, we could evolve into something better. Through progress, we might eventually defeat death itself. Perhaps we might someday even reverse the degradation of the universe. My personal hope was that I might in some small way contribute to such progress. I believe that this basic hope was shared to a large extent by my entire generation." And Dr. Sanford's about 72 years old, just to give an idea of his generation. "One of my reviewers told me that the message of this book is both terrifying and depressing. He suggested that perhaps I am a little like a sadistic steward on board the Titanic, gleefully spreading the news that the ship is sinking. But that is not correct. I hate the consequences of entropy, degeneration. I hate to see it in my own body, in the failing health of loved ones, or in the deformity of a newborn baby. I find it all absolutely ghastly, but also absolutely undeniable. Surely a real steward on the Titanic would have a responsibility to let people know that the ship was sinking, even if some people might hate him for it. I feel I am in that position. Responsible people should be grateful to know the bad news so that they can constructively respond to it. If we have been putting all our hope in a sinking ship, would it not be expedient to recognize this and abandon the false hope? It is only in this light that we can appreciate bad news. Only in the light of the bad news can we really appreciate the good news that there is a lifeboat. Even as we cannot create life, we cannot defeat death. Yet I assert there is one who did create life and who designed the genome. I do not know how he did it, but somehow he surely made the hardware, and he surely must have written the original software. He is called the author of life, Acts 3:15, NIV. I believe the author of life has the power to defeat death and degeneration. I believe this is the good news. It is my personal belief that Jesus is our hope. I believe that apart from him, there is no hope. He gave us life in the first place, so he can give us new life today. He made heaven and earth in the first place, so he can make a new heaven and earth in the future. Because he rose from the dead, we can be raised from death, even the death which is already enveloping us. In these profound yet simple truths, I believe there is a true hope. I believe this hope is unshakable because I believe it is founded on the one who is eternal. It is a hope that has withstood the attacks of time and the corruption of religion. It is a hope freely available to anyone who would receive it today. I humbly put before you this alternative paradigm for your consideration. Jesus is our one true hope. Amen. Peter, could you
Yes.
close us in prayer, brother? And we'll shut
Certainly.
the stream down.
Yeah. Father in heaven, we thank you for this evening that we've had to discuss your creation together. We thank you for the ability to know you and your Son, for whom we have salvation. We ask that you would bless us and also those who are watching this evening, and that you would draw near to those who are in sin and those who need you, and that you would bless us all, and that you would fill us with your Holy Spirit and enable us to love you and to advance the truths of your gospel. Amen.
Amen. I'd like to thank all the viewers, and Peter, we can talk a little bit, and Emery just in the backstage, and that'll do it. I had a great evening. Take care, everyone. God bless you all, especially our new viewers.